Welcome back to the CD Genomics Next Generation Sequencing video series. In this video, you will learn the principle and workflow of the whole genome bisulfite sequencing. This video falls into three parts: overview of whole genome bisulfite sequencing, principle of whole genome bisulfite sequencing, and workflow for whole genome bisulfite sequencing. Whole genome bisulfite sequencing, abbreviated as WGBS, is a well-established method for detecting methylated cytosines at the global genome level. WGBS applies a combination of bisulfite treatment and next or third-generation sequencing technologies to study genome-wide methylation at a single-base level. It assesses the methylation status of almost every CpG locus, including intergenic gene deserts, partial methylation domains, and remote regulatory elements. It reviews not only absolute DNA methylation level, but also methylation sequence background. However, the conversion of unmethylated cytosines into thymines makes it difficult to align sequences. Besides, WGBS is unable to distinguish between 5-methylcytosine and 5-hydroxylmethylcytosine. WGBS has been widely applied in a range of fields. First, stem cell applications. Stem cells are very important for therapeutic purposes and the study of human diseases. They differ from somatic cells in epigenomic transformations rather than genetic ones, making it highly interesting to study their methylation pattern. Second, developmental biology. DNA methylation plays an important role in the development of mammals. In particular, non-CG methylation is widespread in pluripotent stem cells and oocytes. Third, early diagnosis of diseases. WGBS can be used to detect aberrant methylation by screening specific hypermethylated suppressor genes as seen in cancers such as gastric cancer, acute promyelocytic leukemia, and so on. Fourth, forensic science. The use of WGBS improves analysis of DNA methylation on forensic samples. WGBS has become the standard profiling method in major epigenome consortiums. The most commonly used genome-wide DNA methylation profiling methods are affinity enrichment, restriction enzymes, and bisulfite conversion-based methods. As seen in this table, bisulfite conversion-based sequencing provides more reads, covers more CpGs, and is suitable for high-resolution studies. Bisulfite converts unmethylated cytosines in the genome into uracils, which are then replaced by thymines during PCR amplification. Unmethylated cytosines can be distinguished from the cytosine originally modified by methylation by counting cytosines and thymines for each position after sequencing. The workflow for whole genome bisulfite sequencing generally includes DNA extraction, bisulfite conversion, library preparation, sequencing, and bioinformatics analysis. 1 to 5 mg of tissue samples are collected from humans, animals, plants, or microorganisms to isolate high purity and high molecular weight DNA. Studies have shown that once the number of CpG sites in a region increases, the sequencing data of conventional WGBS begins to decrease. Therefore, conventional WGBS is suitable for hypomethylated genome analysis. BS-induced DNA degradation may lead to depletion of genomic regions enriched for unmethylated cytosines. Therefore, it is important to assess DNA degradation, and how this affects the desired amplicon should also be considered. There are several excellent kits available in the market for bisulfite conversion. Take the epigenome methyl kit as example. Bisulfite-treated single-stranded DNA is random primed, using a polymerase capable of reading uracil nucleotides. to synthesize DNA containing a specific sequence tag, the 3' end of the newly synthesized DNA strand, is then selectively labeled with the second specific sequence. Thus, a 2-marker DNA molecule with a no sequence tag at the 5' and 3' ends can be obtained. Illumina P7 and P5 adapters are subsequently added by PCR at the 5' and 3' ends prior to DNA sequencing. Illumina high-seq sequencing technology, based on sequencing by synthesis, is widely applied for WGBS. Besides, PacBio and Oxford Nanopore instruments are also used for DNA methylation profiling. They do not need PCR amplification or chemical labeling of the DNA. The WGBS data are generally processed with several steps, including adapter trimming, a quality assessment of reads, sequence alignment, and methylation calling. After methylation calling, the average methylation level and genome-wide methylation level are calculated and plotted, and differentially methylated regions are determined and annotated through integrative analysis. Thanks for watching. If you want more information about the whole genome bisulfite sequencing from CD Genomics, please visit our website at www.cd-genomics.com. We are more than happy to be of assistance.